Yeah, good morning everybody. I'm working in the Health Directorate of the European Commission, and my responsibility is to promote personalized medicine and to develop funding programs, in particular for human microbiome research, and to be able to justify the funding what we currently want to do. I had to do a portfolio analysis in the area of microbiome research to see what we have funded in the past and what we should do in the future. And I can tell you about the findings from this portfolio analysis. So I will talk about one very important breakthrough project. They came up with the Nature cover page in 2010. And then I want to mention the developments and trends, what I've seen in this analysis. I talk about the challenges and the conclusions. And then finally, I come to three new projects that should normally start in January next year. I hope everything goes well, but we decided to fund three new projects for next year. So the first one was a very important, or the most famous, maybe the most famous metagenomics project that we had. It's a MetaHit project. They came up with this Nature cover page in 2010. Because they discovered a lot of microbial genes and also functionalities, and the most important finding was that they discovered three distinct enterotypes. So everybody of us should have one of these enterotypes, and this is dominated by one specific bacterium. This is what they proposed. And this is independent from which country you are coming and what you are eating, and they compared it with blood types, and this was so exciting at that time that they got a Nature cover page. They also said that the diversity of microbes in the gut is very important, and this is linked to obesity, so it's more healthy to have more different microbes in your gut. And they established the IHMC, the International Human Microbiome Consortium, because they realized already that it's a very big exercise to find out everything about all the microbes and to discover all their functionalities. We can't do this within one project, and we need different countries to participate, and that's why we started cooperation with the US, Japan, Australia, and everybody involved in this big undertaking. MetaHit went from 2008 to 2012, and afterwards we continued with MetaCardis. The focus was then on cardiometabolic diseases. They also had a very systematic approach. They looked at many other factors as well. So they looked at lifestyle, clinical data, drugs, and so on. The project is still going on, but there is already some interesting papers from this project about obesity and also the low gene richness and functional pathways leading to obesity. And in the end, one recent paper is about the bariatric surgery. They say that this is a good first step to do for very obese people, but it needs to be complemented because these people have a very low gene richness in their gut, and we have to find a way to increase this gene richness. So now, the developments and trends in this area. Of course, in the beginning it was very exciting to look at this diversity of 100 trillion different bacteria, and we had to find their genes. It was a very huge exercise. And at that time, metagenomics was still at an isolated off-site position. We would say we had on one hand people dealing with human genetics, and on the other hand people doing metagenomics. And the experts on human genetics said that the gut microbes, this is something apart. It does not really belong to the human being, and they didn't want to take this into account very much. This was some years ago, and this has changed now. Of course, it was important to get solid analytical technologies first, and this took some years. And then we saw an expansion of metagenomics in many other areas. You can say it was an expansion or explosion of metagenomics. We have it now in all areas. For example, also we have projects on the bee gut microbiome, and they also released a paper a few days ago. where they found out that certain pesticides, glyphosate in particular, has an effect on the gut of bees, and then this is modified, and the immune system collapses, and this can probably explain the mortality of bees. It was just released a few days ago. And then there is, of course, projects dealing with the gut microbiome of cattle, and also we have some news on oysters, because oysters mortality was also something very interesting, and we didn't know why they are dying suddenly in such huge quantities, and it's also related to microbes and viruses at the same time. So we had a lot of hope. Suddenly we had a lot of hope on the microbiome research, and we wanted to find out more for health from the microbiome, and human genetics became a bit boring. We couldn't explain that much anymore with human genetics. So, but still I want to emphasize here this last point. We have a trend in health research which is very important. You can see this in all the topics what we want to fund this year, next year, and in the future. We want to find trends in a set of big data. And here metagenomics is one part of the exercise, and we want to take all kinds of other data into account at the same time. This is the most important trend at the moment. So here I have a very modern project. SysKit started only last year. They deal with chronic inflammatory diseases, and you can see, it's a bit difficult to see here. On the side there is a picture from the SysKit project, and you can see different information layers. They have a very systematic approach, and they say that metagenomics is only one information layer, and we have to deal with other epigenomics, for example, and human genetics and lifestyle, nutrition, and we have to take into account everything at the same time. There is still a lot of hope on the microbiome, on the gut microbiome research, but we want to take all information at the same time. Interesting findings are that Western diets are linked to inflammation, and also they found out about the maternal microbiome, which is very important for the unborn baby. In fact, the microglia that is in the brain of the baby is already affected before the birth by the gut microbiome of the mother. Another very interesting project is Eat to be Nice. It's more about behavior, and taking also into account different confounders, and looking at very different parameters at the same time. In the end, they want to promote policy changes. Also very interesting, but started only recently. This gives an overview. I don't want to mention all the figures, but we have quite a long list of projects. We have 216 projects so far for nearly 500 million euro, and you can see there is many in health, but also in non-health there is quite a long list of projects. For example, the built environment is getting more and more important. The built environment is everything what is constructed by humans. So, for example, buildings, houses, rooms, meeting rooms. Also, the ventilation system in the meeting room is very important to look at, because it can influence our microbiome. Here is some more very interesting projects. It's a long list, I don't want to mention everything. So we have projects on allergies, on skin, asthma, and so on. I don't have time to mention all that. The challenges, what we have seen, of course, the big vision in human health is to modulate health via the microbiome. This is a great idea what we have, and a lot of companies are getting ready to discover some kind of drink or yogurt that contains certain microbes, and then this could modulate the gut microbiome and influence our health or disease state. This is what we hope. But we also see at the same time that the mechanisms are more complex. Now, over the last 1 or 2 years, we have seen there is too many confounders that need to be taken into account. It doesn't go so fast that we can have these drinks or yogurts on the market pretty soon. We have to take into account all kinds of other confounders, multi-omics, lifestyle, drugs, the region where we are living, and so on. And we also have to see that there is a competition among microbes, and they change and adapt very quickly. And this we also need more research on this. And there is an interplay with the environment. I said, for example, the meeting room could be. We could have an interplay. There is transmission between people when we shake hands, or when the ventilation system has certain microbes. This is something what we have to look at. What is also very important for health research is to get bigger cohorts. Here's, for example, I would like to mention a Nature paper just a few days ago on the UK Biobank, where they really work very hard to collect data from 500,000 people. We have the same initiative at EU level. We want to collect data from 1 million people. You maybe know we launched in April the initiative, the 1 Million Genome Initiative, where we collect data from more than 1 million people by 2022, just to get comprehensive information and to be able to see some trends for diseases or health states. What is also very important, of course, to get harmonized methods and standards, especially for microbiome research. The taking of samples, the storage, and the data processing is done differently in different projects. And we have a problem afterwards. We can't compare data from different projects. The conclusion is, how do we want to promote personalized medicine in future? It's very important not to have data silos, not to work with one discipline only. We need multidisciplinarity, and we want to integrate data from different fields. There was also a Nature publication a few days ago saying that we must involve people who hope to benefit from our research, and this is patients. So patient organizations should be involved in our projects. This is then more difficult, of course, to work with them, but in the end there could be greater results. And we want to move from reactive to proactive approaches, so more looking towards the prediction and prevention. By these conclusions, we ended up with the topic BHC3, what we published, and there was a deadline this year in April. We got 27 applications to analyze. We have analyzed all these applications, and we have finally decided now during the summer what we're going to fund. And I can tell you the 3 projects that we want to fund and that should start in January next year. The first one is called Microbiome. It's for 15 million euro. It's looking at cancer. Actually, they want to make a cancer microbiota atlas. They have very large cohorts dealing with more than 9,000 cancer patients across 10 countries. And they want to predict the treatment response, of course, and influence cancer progression. So in the end, they want to make a companion diagnostic test. This project is coordinated by Laurent Sitvogel. The second project that we want to fund is called MicroPredict, also for 15 million. And this is about the liver failure. And they also deal with a very big cohort of more than 10,000 patients. They want to find the functionalities leading to this disease, and they also want to come up with a clinical decision support tool. This should be validated within the project. It's a long exercise, will take 6 years, but this is of course a high impact outcome, what we hope if we get really tools for clinicians or end users. This was the main purpose of our topic, to get such tools. And they want to have also at the same time for the end users an easy to use nano biosensor. This is a point of care device for the end users that is connected to smartphone, to the smartphone of the end users, where they can see, where they can learn about their health state. So this is a high impact application, and this is one of the reasons why we want to fund it. And I said We want to see patient organizations involved in the work. This is very difficult because they are living in a quite different world, but here we have a patient organization involved in the project, and they are going to disseminate the results, so translate the results of the project to the general public. This is something of great added value, and we hope to see more projects like this in future. The last project is called Gamma, also 14 million euro, and it's about autism. And there is an increased rate about autism. What you can see over the last years, it's going up extremely, and that's why we expect also high impact from this project. They involve more than 600 children at risk. They want to understand the mechanism that is behind this autism. And in the end, they want to come up also with an analytical platform that is using artificial intelligence and multi-omics to make some predictions for these patients. And they also want to design a preventive nutritional formulation. So that's it in principle for me. We have 2 topics open at the moment. The deadline was already 2nd of October. I don't know, it doesn't make sense to talk a lot about these projects, but it's about understanding the mechanisms behind co- and multimorbidities. And the another one is about demonstration of personalized medicine in real life. That's very exciting. It's also about finding trends in big data. And metagenomic data belong to these data, but also all kinds of other data are taken into account at the same time. So for the future, we want to link different actors, bring them together. Multidisciplinarity is very important, involving end users, citizens, and as I explained, we want to explore the multitude of big data and to find trends here. Standards are very important for data comparability. We have to work here. The technologies are developing so fast that we are not able to develop the standards at the same time. And it's about real-life implementation of personalized medicine approaches. And of course, digital tools. We want to see digital tools on the markets. This is what we want to fund in the next years. We have not decided on the final topics for the next years. Horizon 2020 is finishing, and Horizon Europe is at an early stage, is still to be decided. But this is probably the most important things, what you will also see over the next years in our topics. Thank you.
Thank you very much, Dirk. Any questions?
Wolfgang Ahrens, University of Bremen, and Leibniz Institute for Prevention Research and Epidemiology, Bremen. You mentioned the UK Biobank, and you mentioned these huge projects that collect data, that collect genetic material for analysis. Now, the UK Biobank is not EU funded. And these kind of studies, similar studies, are ongoing in other countries. Like in my countries, it's called the National Cohort. There's Lifelines in the Netherlands, and there's a similar study in France, all using kind of different protocols. These studies are not harmonized. But these studies are very important because they are longitudinal studies, and the duration of these projects, because these are cohort studies, goes far beyond the usual funding period of 3, 4, 5, 6 years that is possible within EU framework programs. So my question is, shouldn't the Commission think of establishing something like a pan-European cohort with a funding scheme that goes beyond the usual project duration? Because these projects, if you establish these kind of projects, you have to think of 30 years of follow-up of these subjects in order to investigate pathways and the causal development of disorders in relation to prospective data.
Yeah, yes, of course, you're right. We need a longer process to investigate all this. I mentioned the UK Biobank with 500,000. because they were pushing for this exercise, and they started the exercise, and then EU took over, and now more and more countries are getting involved in this, and we have this EU initiative to collect 1 million genome data by 2022, and we are only at the stage where more and more member states join this initiative. So in the beginning, I think in April there were only 13 member states in favor, but now more and more, I don't remember the last update, but more and more member states joined this and also signed the declaration to do this. And of course then the next step is to define parameters for this big exercise, but of course it's very difficult the more member states are joining. We also need a lot of discussion how we collect the data, how we process the data, and the relevant standards for this will not be easy. But the initiative has started in April, and we are not that advanced. There was a first meeting in September, and there's another meeting in December to talk about this, what you mentioned, to develop standards and to get further. Yeah.
Respond briefly?
Yeah.
I mean, the genome is only one thing. You need not only genomic data, but you need phenotypic data, like health behavior, for example, and physical activity, diet, all these information. So the genome is not enough. And then you need longitudinal collection of data, and you need health outcomes. You need to observe health outcomes later in life. That's important.
Yes, I fully agree with you. It was in the knockout. We had a knockout consortium some years ago, and they were only looking at knockouts. And then afterwards, they were more interested in the phenotyping. This is the second step. We can't do 2 steps at the same time, and we are not very fast because there is so many member states involved, and we need to agree the way we go. And I'm sorry, it doesn't go so fast, but I fully agree with your ideas. Yeah.
All right. Thank you very much. And this is no doubt a very interesting conversation to have. And we can continue over coffee breaks. And I know I saw more hands, but I'm very sorry. I think we need to continue with the session.